The overall goal of this experiment is to develop a simple LAMP method using lyophilized reagents for the detection of C. burnetii in patient samples.
The main advantage of this protocol is that no cold chain is required for the storage of the reagents. This method is ideal for use in resource-limited settings where Q fever is endemic. Demonstrating the procedure will be Tatiana, a technician from my laboratory.
Beginning with DNA for the target gene IS1111A from C. burnetii RSA493, determine the plasmid concentration with a spectrophotometer at 260 nanometers. Convert the plasmid DNA concentration into copy number using the following calculations, where 6,330 equals the length and base pairs of the plasmid, and 34.2 nanograms per microliter is the concentration of the plasmid, as just determined using the spectrophotometer. Then prepare 8 tenfold serial dilutions of the plasmid to arrive at the following number of copies per microliter. To prepare the DNA template for the LAMP reaction, begin with 200 microliters of human plasma samples, and with the DNA mini prep kit, extract the DNA according to the manufacturer's protocol. Elute the final sample into a 20 microliter volume. Prepare 1 milliliter of 2x LAMP reaction buffer by combining the following reagents. Mix by pulse vortexing for 10 seconds. To perform the standard LAMP reaction,